Hi, everyone. This is Janet Dafoe here, and we're recording the fourth in my series of interviews of the work done in Ron's lab. And this one isn't technically done in Ron's lab, but it's collaboration with Ron. And I'm so pleased to have Rob Phair here, who's been collaborating with Ron for a long time and is just doing really exciting work along with Ron and with Chris Armstrong in Australia. So I do know that Ron and Rob talk very often every week, and that hour on Thursdays with Rob is Ron's favorite hour of the week, except with the possible exception of his hours with his grandchildren. So I'm really pleased to be doing this interview, and we're real excited about this work. Okay, Rob, if you can go ahead, and then when you're ready, you can share your screen.
Okay, I'll do that. Thanks.
Mm-hmm. Thank you so much.
All right, I'm happy to be here too. This is my second hypothesis for ME/CFS. Many of you have heard my IDO metabolic trap idea. That one was supported by OMF, and this one is supported by the Amar Foundation. That's Vinod Khosla's foundation. And the work, I thought I'd start by telling you the story of how this project got going. Back in 2019, just before the pandemic, Chris Armstrong was here in the Bay Area visiting Ron's lab. And during that period, Chris and I got together every Thursday to talk about metabolism. And you'll all remember that Chris originated what's called the ammonia hypothesis for ME/CFS. And at the time, we talked about his data, which show that amino acids are being oxidized in CFS patients in preference to carbohydrates and fat. And so during that summer and fall of 2019, Chris and I talked about two questions. One was, what could cause this kind of change? What could result in the burning of amino acids instead of, say, sugar? And the other question was, what makes it chronic? So then you all know what happened at the beginning of 2020. We started shutdowns, and Chris went back to Melbourne. The lab shut down at Stanford. And so I volunteered on a different project on COVID-19 for a good period of time in the early going of the pandemic. And I learned a lot about innate immunity while working with this team at the Luxembourg Center for Systems Biomedicine. And it turns out that that knowledge about innate immunity that I gained by working on that project has come to be important in my ME/CFS research. And so at the beginning of 2021, Chris and I presented a proposal to the Amar Foundation on traps or bistabilities, which are kind of like on and off switches in amino acid metabolism. And that proposal was funded by Vinod Khosla. And we started working on it right away. And by May of 2020, I had concluded from my modeling that the itaconate pathway that I'm going to show you today was our best lead as a possible cause for the switch to amino acid metabolism. And as Janet says, I talk to Ron every week. And so I mentioned this to Ron, and he was so enthusiastic. He's always been a great supporter of mine, but he was so enthusiastic that he shifted some of his really good people at the Stanford Genome Technology Center to begin looking at how we could measure things that would allow us to test the ammonia hypothesis or the Itaconate hypothesis.
So I hope you're going to mention your favorite book here.
Okay, well, I said the last time I talked to Janet about this, I remembered that you can't test a theory without experiments. And right here is the point where I made that point about a book called The Knowledge Machine by Michael Strevens. And it's a book about the philosophy of science. And Janet eventually read that too, and now considers it one of her favorite books. So the interplay between theory and experiment has been a big part of my scientific life. And that's what we're doing right here. I've developed a theory, and Chris and Ron will both be able to look at the predictions of that theory and make measurements to test it. Okay, so those people that Ron moved part-time to this project are listed here. And the tool that I use to do my modeling is called ProcessDB, and it was developed in our company, Integrated Bioinformatics, by two really good computer scientists. Ann Chasten is a developer of databases. She worked for Oracle for many years. And Jason Zwolak is a world-class software developer who also happens to have a PhD in computational cell biology. So that's the group that's working on this. And by September of 2021, we had an answer to the first question. The first question being, what could cause the change to amino acid metabolism? And that's what Janet invited me to talk about today. So I'm going to do that. And if I do a good job, maybe she'll invite me back to talk about the second question, which is what makes it chronic? And that's what we're working on here in 2022.
I'm definitely inviting you back.
You are? Okay. Well,
yes.
See, and she doesn't even know whether I've done a good job yet.
I'm sure you have.
Okay, so this is a complex biochemical system. I'm going to go slowly. I'm going to go step by step. I'm going to show you the models actually in ProcessDB as we go. So this is the first model. And it's a model of what is called the tricarboxylic acid cycle, a TCA cycle. Many people know it as the Krebs cycle in honor of Hans Krebs, who delineated it back in the 1930s. And I wanted to first say a little bit about the glyphs on the screen. That is, the diagram itself represents a set of biochemical reactions. And so each arrow, here, all the arrows represent enzymes that are catalyzing conversion of one molecule to another. And all the rectangles represent molecules, or what we call states, because they're molecules in a place, and the place is the mitochondrial matrix. So this is the Krebs cycle running in the mitochondrial matrix. A lot of
excuse me, mitochondrial matrix, does that mean inside mitochondria?
Yes, it's the fluid phase inside the inner membrane of the mitochondria.
Thank you.
And so a lot of beginning biochemistry students are puzzled by this cycle because it appears to do nothing. It just converts citrates. This is a conatate, and so forth around the cycle. And eventually you come back and you have citrate again. So what's the big deal? And of course, the answer is that this is the center of carbon metabolism in the human body. And one way I have found it be useful to explain that cycle and why it's so important is to think about its inputs and its outputs. And so now I'm going to add to this diagram the the inputs from the outside of the TCA cycle, and in fact, outside of the mitochondria. And so these green arrows that are now added to the diagram, one glycolysis, delivers the consequences of eating sugar. So glycolysis is a process that converts glucose eventually to pyruvate. And pyruvate enters the mitochondria through a carrier that is specific for pyruvate. And then pyruvate can be converted by an enzyme called pyruvate dehydrogenase into acetyl-CoA. And acetyl-CoA can combine with oxaloacetate from the TCA cycle and make citrate. So that's how glucose gets in. And this second process in green, beta oxidation, is the process by which fatty acids get into the Krebs cycle. And so those two macronutrients, sugar and fat, get into the Krebs cycle at the same point at acetyl-CoA. Down on the right-hand corner, lower right-hand corner of the diagram, you see two processes by which amino acids can get into the Krebs cycle. And so what's important here is that the inputs to the TCA cycle are the nutrients that we eat. So they're sugar over here, fat up here, and amino acids down here. So now we want to ask, what are the outputs of the TCA cycle? What makes it important for the cell or for the human body as a whole? And those outputs are indicated by the red arrows in the TCA cycle. At each one of those red arrows, something is made that allows the cell to produce energy. At all of the dehydrogenases, you get reducing equivalents. So you get a molecule called NADH, or you get a molecule that's called FADH2. At the STK, you actually get ATP itself, and at the other two, you get NADH again. These NADHs are used by the electron transport chain in the mitochondrial inner membrane to make ATP. And so the importance of the overall diagram, the overall model, is that fuels come in and ATP comes out. Probably you all know ATP is the energy currency of the cell, and it's used to do all the other work that cells must do, like pumping ions across membranes and synthesizing proteins. So now what we're going to do is add to this picture the Itaconate shunt. And this is not something that's normally operating in the mitochondria. Instead, I'll do that here. We add the Itaconate shunt, which is initiated by the innate immune system. This comes back to my year working on COVID-19. And the innate immune system triggers the production of many enzymes and also many transcription factors. And I will talk about this more in detail when Janet invites me back. But the key here is that one of the things that a viral infection or a bacterial infection or an injury, any kind of trauma, the innate immune system will activate many things, and one of them is the production of this enzyme called CAD, which stands for cis-aconate decarboxylase. So it removes the carbon dioxide from cis-aconate and creates itaconate.
So Rob, I'm sorry to interrupt you here, but could you say just your little blurb about innate versus not innate immune system?
Right. The innate immune system is the first responder to an infection. It takes perhaps 4 days or somewhere between hours and 4 days to complete its job. And then it hands over the response to the infection to the adaptive immune system. The adaptive immune system is the one you've mostly heard about. It's the
Some people call it acquired. Is that the same thing?
Could be. I don't know.
Okay, sorry.
And so it is in a sense acquired, so maybe it is. That's the T cells and the B cells. The B cells, of course, produce the antibodies. All of the things you read about in the newspaper for COVID-19, for example, are part of the adaptive immune system. The innate immune system is first. It's the ancient immune system. You can go back to single-celled organisms, and they all have an innate immune system, whereas the adaptive immune system is only present in vertebrates, like us. Okay, so back to CAD. CAD is an enzyme. It catalyzes the conversion of itaconate to itaconate. Itaconate, by the way, is an anagram of the letters in aconate. I don't know why they did that. To me, it's kind of confusing, but that's the way they did it back in the 19 whatever. Okay, the second step in the itaconate shunt is catalyzed by one of the enzymes of the TCA cycle. That's succinate thiokinase, STK. And it's actually running backwards from the way that it runs in the normal Krebs cycle. It's putting CoA on itaconate, making itaconyl CoA, whereas in the normal Krebs cycle, CoA is being removed and producing an ATP. That means this one is actually consuming an ATP. Okay, so now you have itaconyl CoA, and another enzyme in the mitochondria converts that to citromylyl CoA. And up till 7 years ago, this was a dead end. No one knew what happened next. They thought they were just building up these CoA-containing molecules. But 7 years ago, a group at Harvard in Vamsi Mutha's lab de-orphaned an enzyme called Clibal. It had been known, people knew that it was like lyases. And it turns out that it is a lyase, but we didn't know what it would do because we didn't know what its substrate was and so forth. So this Harvard team figured out that citromylyl CoA is broken down to pyruvate and acetyl CoA. And that completes a kind of cycle. It's not the normal Krebs cycle, but it does get you from having made acetyl CoA going around, you go around this cycle, and you come back and you have pyruvate and acetyl CoA again. So you can start over again. But the question is why? Why would you do this? Now you've skipped all these red processes. You've come out of cis-aconate and get back to pyruvate, but you haven't done any of the energy making steps except the first one over here at PDH. So you're only making one NADH instead of many NADHs.
So that's why it's called a shunt, right?
That's why it's called a shunt.
Shunting away from the normal Krebs cycle process.
Yes, that's why it's called a shunt. And so why would the body do this? Why would the innate immune system want to do this? Well, the idea is that it's not only preventing production of energy for the cell, but it's preventing the production of energy for the virus or the bacterium. And so the idea here is that the cell believes that it can stave off the bacterial or viral infection long enough to hand off the responsibility to the adaptive or acquired immune system. And so this is just a mechanism for slowing the ability of the virus or the bacterium to reproduce inside the cell by restricting its access to the very same energy-containing molecules that the host cell uses for its own purposes. In addition to that energy stealing response to the virus, there are two steps indicated by this. Actually, I only have one of them here, but the itaconyl CoA is an inhibitor of the last step in getting branched chain amino acids into the Krebs cycle. And itaconate itself is an inhibitor of succinate dehydrogenase. So everything about this pathway is an attempt by the innate immune system to restrict energy production in this particular cell that's infected. And so now the consequences of this pathway need to be addressed, because the hypothesis that we're getting to here is that this pathway, the itaconate shunt, is on chronically in an ME/CFS patient. That is, at some point in the initial infection, this pathway was turned on and it has not yet been turned off. And that's the main theme of the second half of this, which you'll hear whenever we get to that in the next month or so. So now, if you have this pathway running in a steady way, in a chronic way, what happens is that the enzyme CoA is a coenzyme, which is made from a vitamin by a pathway that is here, but that's another long story. You can make CoA slowly from a vitamin, but if it's building up in these two molecules, itaconate-CoA and citramyl-CoA, then you have sequestered the CoA molecule away from the rest of the reactions in the mitochondria. And so this slows pyruvate dehydrogenase, which requires CoA to make acetyl-CoA. It slows beta oxidation of fatty acids, because you have to have CoA. For every 2 carbons in your fatty acid, you have to make an acetyl-CoA. And so again, you need CoAs again and again and again. And finally, you are slowed in this final step of the branched chain amino acids entering the Krebs cycle, because again, you have to provide a CoA. So you can see immediately that if you slow glycolysis and slow beta oxidation and slow branched chain amino acids entering the Krebs cycle, then the only source that's left is glutamate coming in at 2-oxyglutarate. This is the only way into the Krebs cycle that does not require CoA. But there's a problem, and that problem is that glutamate getting into the Krebs cycle at 2-oxyglutarate, that's a good thing. But the problem is that 2-oxyglutarate dehydrogenase, the step that's going to complete the movement to succinyl-CoA, also requires CoA. So you can't get from 2-oxyglutarate to succinyl-CoA. You have nowhere to go. And so that leads us to the next stage of this talk. If you don't have anywhere to go, you have to rely on the flexibility of carbon metabolism in the human body, or in fact, in most living systems we know about. And that flexibility allows a working but not very efficient alternative TCA cycle. And so that's what I'm going to show you now. This is what's left of the Krebs cycle when you have sequestered all the CoA. Now, it could well be, of course, that some of the carbons going into the Krebs cycle are still going by the usual route. Not all of them are going to go by the itaconate shunt. Some of them will still go on to isocitrate and complete a normal cycle. But to the extent that CoA is sequestered away from the TCA cycle, you're stuck with only these reactions that are on the screen now. So you can get from succinate to fumarate to malate and back to oxaloacetate, but you have no way to complete the cycle because the remaining steps can't run without the CoA molecule. And so I mentioned a moment ago the flexibility of carbon metabolism in human biology. And that one of the most beautiful examples of that is what's called the GABA shunt. And so here's a set of 4 enzymes that are already present in the mitochondrion that can complete the gap between oxaloacetate and succinate, even in the absence of CoA. So here we go. This first enzyme is one of Chris Armstrong's favorite enzymes. It's a glutamate oxaloacetate transaminase, GOT2 is the gene's name, and it allows the conversion of oxaloacetate directly to 2-oxoglutarate by just moving an amino group from one place to another. Now you can do 2 things. You can convert that glutamate, or a glutamate, you don't need to use can't use the same one, but you convert glutamate as well to GABA. GABA stands for gamma aminobutyric acid, and the enzyme is called glutamate decarboxylase. So you lose a carbon dioxide here and you get a GABA. Now you can do another transamination. You can move another amino group, move one back to glutamate, and put and then get succinic semialdehyde from the GABA carbons. There is an enzyme called succinic semialdehyde dehydrogenase, and that has 2 great advantages. One, it actually makes an NADH. So you can now use that NADH to make ATP. And in addition, it completes the circuit to succinate. So in effect, you had, when you remove the CoA by putting it into a itaconate-CoA and citrumallyl-CoA, you had an open circuit. You blocked the pathway from oxaloacetate to 2-oxoglutarate and then to succinate. But this flexibility of carbon metabolism, the GABA shunt it's called, you can see how it's also a shunt. It's a completing of a pathway around this broken loop, is now complete, because now you can convert, you can move carbons from oxaloacetate all the way to succinate and complete the loop. It's not a very efficient loop. We calculated it's about 43% of what you could get if the Krebs cycle was running at the.
So does it create ATP somewhere in there?
Yeah, so NADH, as I said on the previous diagram, is used by the electron transport chain. You don't want to see that diagram yet. That's a really complex diagram.
Oh yeah, no.
Okay. All right. So NADH being produced by this dehydrogenase is one of the molecules that can be used to make ATP. And there's also the one that's created from malate dehydrogenase, making NADH. And you'll remember, although it's not on this screen, that when you get from pyruvate, well, you can't do that anymore because you don't have CoA. You can't get an NADH from pyruvate anymore. You can only get pyruvate in through 2-oxaloacetate.
And what are the inputs to this shunt? Is it the lipids, the sugars, or the amino acids?
It's the amino acids. So it's
and that's basically protein, right?
That's what proteins are made of.
Yeah.
Proteins are long chains of amino acids, and glutamate is a very common intermediate in the conversions among various amino acids. In fact, GOT2 is in effect converting oxaloacetate into aspartate in exchange for making 2-oxoglutarate. So a lot of amino acids can be converted to glutamate, and glutamate can get into the Krebs cycle by converting to GABA.
So let
Glutamate,
me just do something here, because I'm imagining some of the people watching this are getting their eyes glazed over because there's a lot of terms and things in here that are hard to remember, and you don't know what they are. So just in general, the normal Krebs cycle makes energy in your mitochondria, and you operate fine with it, and you get a viral infection or some other trauma, and it triggers this itaconate shunt, which makes it really hard to process sugars and lipids and amino acids. And however, there's another shunt that happens that allows you to make it energy a little bit using amino acids.
that's correct.
Okay, that's just for people. And let me just encourage you who are watching, just let some of the details, if it's too much, just let them flow past you, because at the end you'll have an idea of what this is about, even though you couldn't possibly repeat all this stuff that Rob is saying. It goes together to make a beautiful picture.
Right. I think it's beautiful.
Yep.
We'll let the viewers decide for themselves.
Yeah. I just don't want them to give up, because
Oh, okay.
yeah.
Well, I don't want them to give up either.
Right.
Okay, so here we are. We've got a 43% efficient Krebs cycle that's been created by the magic of the GABA shunt, and it's burning glutamate in the form of GABA to put carbons into the Krebs cycle, create a little bit of NADH. It also creates an FADH2, which is also capable of making ATP. So you have three reducing equivalents that are being made by this modified, rewired. Rewired is the term you see in the literature these days, rewired metabolism. But it turns out, and very importantly, that the lack of energy is only the tip of the problem iceberg. This problem goes well beyond the robbing of energy that could be used to make ATP. And I thought I'd give an example of how that works. And so the example I chose is the brain. And so we move on to this next diagram. And I'm going to start by saying a little bit about how the brain normally works. You probably are all aware. So let me say something about the diagram first, because this is the first diagram that has more than one place in it. So all these molecules on the left are in a glial cell in the brain. And then there's a space between cells in the brain. It's called the extracellular fluid. Some of the extracellular fluid is actually in the synapses, which are the place where one neuron signals to another neuron, where information is transferred. And those neurons are basically of two major kinds in the brain. There are what we call glutamate neurons and what we call GABA neurons. Glutamate neurons are the excitatory ones. They send messages that depolarize the next neuron and say, send this signal on further down the network. GABA neurons are the major inhibitory neurons of the brain. And they send messages that say, don't send as many signals as you have been sending to the next. They send that message to the next neuron in the chain. So what normally happens in the brain when a glutamate neuron sends glutamate out into its synapse is that it signals to its downstream neuron. That is, it depolarizes the downstream neuron. Action potentials travel down the neuron carrying information. But that glutamate is still in the synapse. And we don't want to convey old information. That would confuse everything. We want to convey the most current information. So the glial cells, which wrap all the synapses, are charged with the responsibility of taking up the glutamate that's in the synapses and converting it to glutamine. They do this because glutamine has no ability to stimulate downstream neurons. And so it becomes a kind of currency from which you can make neurotransmitters, but which is not a neurotransmitter itself. To do that, you have to put an ammonia group on, or amino group onto glutamate and make glutamine. And that's transported out into the extracellular fluid, not into the synapses actually, but this covers both. And then it is taken up by glutamate neurons. And this is called the glutamate cycle because it allows the glutamate neuron to make another molecule of glutamate. And then it stores that in its post or presynaptic vesicles for delivery to the synapse the next time this nerve is doing its job. The same thing happens for the GABA cycle. Yeah, you transmit messages in the GABA neuron, you release GABA into synapses, and that sends an inhibitory message, but you don't want to continue sending that message. So you take up GABA into the adjacent glial cell, and using one of the reactions I showed you in the previous slide, the GABA transferase, you turn that GABA into succinic semialdehyde, and you use a 2-oxyglutarate to turn it back into glutamate. That can be aminated to make glutamine, and now you have 2 cycles going on simultaneously in the brain, constantly supplying new glutamine from which both excitatory and inhibitory neurons can make their neurotransmitters. So now this is the way it's supposed to work. But the point that I was making in the last slide, that the itaconate shunt causes problems, is not just a causing of energy problems. In the brain, for example, it's going to be causing even more of a problem, because the rewired TCA cycle is now using that glutamate molecule and the GABA molecule to make energy. In other words, because you can no longer use sugar, which is the main fuel in the brain. Most fats are not used by the brain as a fuel. So you can't use glucose because you're lacking CoA, and you need CoA to get pyruvate into the Krebs cycle. So using this rewired Krebs cycle that we talked about 2 slides ago.
That was the second shunt you talked about, the GABA
Right.
shunt.
Yes, exactly.
Thank you.
So now you're using the GABA shunt and the rest of the remaining Krebs cycle to make energy, but you're using glutamate and GABA as fuels to run the Krebs cycle. So you are in effect burning your neurotransmitters. You are using the amino acids, glutamate and GABA to produce ATP, because that's the only way you have to make ATP anymore.
And that's happening in the mitochondria,
Yes,
right? But it doesn't allow it to be used in the brain.
Yeah, exactly. See, Janet has figured this out already.
No, I'm just trying to make it clear to people who can't quite follow all the details, which I'm partly in that category myself.
All right. Well, so there are a lot of details here. That's one of the reasons I split this talk into 2.
It's great to have all the details. It's great.
Right. So you got to have both details and big picture. I tell people, I've told my students for years that in order to be good at scientific research, you have to have a passion for the big picture, and you have to be essentially
OCD
about the details.
Yeah.
Right? Yep. And so the details are in these diagrams, but you can see that the big picture becomes clearer when you have a visual representation of the details. And this is the way we work. This is what I'm showing you in ProcessDB is my daily working space. We convert these diagrams directly into mathematical models, and then we can do simulations on them to test hypotheses against experimental data. So this is just our graphical user interface to the world of mathematical models based on physics and chemistry. We convert all these reactions to rate laws. That's an algebraic form that tells you how much flux, how many molecules per minute are going through each of these arrows.
So you figure out the rate at which each one of these reactions can occur.
That's right. That's the idea. And we figure that out based on fitting experimental data. So it isn't obvious immediately. We've got a lot of information about these molecules, but we don't have all the information. And we certainly don't know how it works exactly in the brain, because all the information we have is based on biochemical assays done in test tubes. So now you see that the big problem in the brain is that you can only make energy in brain cells by using amino acids, because glucose can no longer get into the Krebs cycle. And because you're using amino acids to make energy, you have fewer amino acids to make neurotransmitters. The neurotransmitters glutamate and GABA are both being consumed as fuel. And so you have this problem that is very likely to culminate in what we all call brain fog. This is an inability to do cognitive work, which depends entirely on glutamate and GABA. So this would be bad enough if that was all that was happening. But we think also, Chris and I have talked about this extensively, that glutamate is going to be used not just by GOT2 and by GABA production, GAD. It's going to be used also by an enzyme called GDH. And so GDH is an enzyme that converts glutamate to 2-oxoglutarate. In the normal Krebs cycle, it's frequently used to do just this. But this enzyme is activated by ADP. And so if you are thinking a lot, or if you are doing physical activity, you're going to move the balance of ATP to ADP. You're going to use
What's ADP?
That's adenosine diphosphate instead of adenosine
So
triphosphate.
it's not energy.
Well, it's the byproduct of using energy.
Oh, okay.
Okay. So every reaction, like muscle contraction or pumping ions across the neuron membrane, all of those require ATP, the energy, and they're going to produce ADP. So any of those activities that we think of as inducing post-exertional malaise in ME/CFS are going to convert ATP to ADP. And that's why we think it's important that GDH is activated by ADP. That means that when you do other activities or increase your level of activity, you're going to convert some of the glutamate to oxoglutarate, and in the process, you're going to make ammonia. And so making ammonia is not a good thing because ammonia is a known neurotoxin. And so we think that it's possible that this pathway will explain the post-exertional malaise phenomenon.
Wow.
So this is what I wanted to tell you today.
Can I ask a question?
I'm not done yet. I'm
Oh,
almost
sorry.
done. This is how the Itaconate shunt, driven by the innate immune system, that's the ancient immune system, can turn an infection, which we often think of as one of the many ways that CFS gets going can turn an infection into into CFS. You now have a pathway that's running in the mitochondria that makes the TCA cycle less effective, less energy efficient, that also produces brain fog if it's happening in the brain, and may even account for post-exertional malaise. And we have to test this, obviously. This is a hypothesis. It's an idea. It's a model. And that's why the collaboration with Chris's lab in Australia, he's going to measure itaconate itself using NMR, and he's designing carbon-13 tracer experiments to see whether the TCA cycle is in fact shunting cis-aconitate into the itaconate shunt. And then finally, it raises the question of what makes it chronic. Now, I showed you back here in this earlier diagram that the innate immune trigger is generating CAD, the enzyme that starts the itaconate shunt. And that would be fine, and it's certainly true that that happens, that's well established, but it shouldn't be true in a chronic sense. It should happen for maybe a few hours or at most a few days while the innate immune system is limiting the ability of a virus to reproduce, for example. And it will stop doing this at the moment the adaptive immune system can take over and deliver antibodies to that virus that will stop and clear the infection. So there is something missing here in this theory so far. And that's the topic of the next session, which is how can this shunt, this production of the enzyme CAD and the itaconate shunt, how can that come back to be a chronic element of human metabolism? Why is it that it does not turn off when the innate immune system is supposed to have stopped working? And we have a lot of ideas about that. We have ways that we think we can test some of those ideas. And that's what I'll talk about next time. Okay, Janet, it's all yours.
Does this enable you to predict or the severity levels of different patients?
We'll be able to talk about that with the next session. But I think that if the hypothesis was true, you would suspect that the more cells in your body that are running the itaconate shunt, the worse you will be. The more severe will be your disease.
And then
so we think we can measure some of the molecules in the pathway that we believe will make it chronic. Some of those molecules should be elevated in patients, and they should be more elevated in severe patients than in less severe patients.
And can you predict what would, why some patients would get more severe versus others staying more moderate?
Yeah, I think so, but I only have a few ideas on that. Here's one right here, clival. This enzyme is known as a knockout enzyme in humans. It's not, what that means is that there are multiple common damaging mutations in clival. Some of you have heard me talk about common damaging mutations for the IDO metabolic shunt or trap. And common damaging mutations in clival inactivate it as an enzyme. So there are lots of people in the world with who have heterozygous deficiency in clival. And as a result, citromylyl-CoA and itaconate-CoA will build up to even higher levels because they only have one copy of this gene, which allows you to clear them. So that could be a genetic predisposition for acquiring this this shunt causing disease. And it's another set of things that could amount to predispositions are up here in the pathway of innate immune triggering. There's a complex pathway. I mean, I can show you what we're going to start with next time. That's this guy. This is the pathway. Yeah, this is a really complex pathway. Janet's groaning because now she knows why I didn't put it all in one talk.
Yes.
But this pathway has a lot going on in it that is worth in its own talk. And you see right down here at the bottom is where the production of CAD and the first enzyme in the itaconate shunt is happening. So there can be predispositions in this pathway as well as the pathway we've looked at today.
So then I presume when you're talking about what keeps it going rather than stopping like it does in normal people, you might talk about what could undo it.
Oh, absolutely. Sure. That's why we're in business, of course.
Yeah.
We're in business because we want to cure ME/CFS. And the first step in understanding how to cure it is what is the mechanism of CFS?
Yeah.
And this diagram is a hypothesis about what that mechanism is. And that one, the one that we'll talk about next time, is a hypothesis about how it gets going and possibly how it stays going when it should be turned on. You know, these are bistabilities. I talked about this with Janet some time ago. Bistabilities are like on-off switches. They're like the light switch on the wall. It's got two stable modes. It can be on or off. And there are bistability mechanisms in metabolism and in cell signaling. This diagram is all about cell signaling. There's not much metabolism here. There are transcription factors, there are receptors, there are cytokines, and there are gene upregulations and inhibitions. Those are all what we call signaling. When I was a professor years ago at Johns Hopkins, I did a course on cell signaling systems. And we called it second messengers, because a lot of these molecules in the cytosol of the cell are second messengers. They're receiving signals from cytokines outside the cell, being transduced by a receptor, and making messages in the form of transcription factors that are sent to the nucleus, upregulate and downregulate genes, and then turn on processes like CAD. That's just one of many, but it could be a really important one. And bistabilities in this system would, for example, not turn it off when it's supposed to. If it's supposed to be off by 4 days after the infection and it stays on because one of the inhibitors, like SOX3, we'll talk about next time, is inhibiting the whole signaling system and preventing turning it off. If it's not being turned off, then it'll stay on and you'll get production of CAD even when you don't want to. So we think there's a bistability hidden in this system. That's what we're working on in 2022. And I'll tell you as much as we know about that next time.
I'm a little hesitant to ask this question, but everybody keeps asking it, which is, what is the relationship between this and the metabolic trap?
Well, there is one relationship that I think is intriguing, but when you say the metabolic trap, you mean the IDO metabolic trap?
Yes.
Well, it turns out that interferon signaling, which is what's in this diagram, turns on many things, and one of them is IDO1.
Oh.
So that's a relationship. I don't think that it's necessary for the IDO trap to be working. but they are connected, and they almost have to be, because we all think that the immune system is an important part of the pathology of ME/CFS. And both the IDO metabolic trap and the Itaconate shunt are directly connected to the immune system in the human body. So the interferon alpha out here on the right, the cytokine that's doing the signaling, is an important part of the innate immune system. And the fact that it turns on IDO as well as CAD is, I think, interesting, but it's probably just because the IDO trap is directly affecting the immune system by regulating the AHR. It's the aryl hydrocarbon receptor, which is again sending signals to the nucleus and upregulating genes. So they're closely connected, but I don't think you need to think about IDO in the same context as the Itaconate shunt. There are two hypotheses, both of which could be true. And the one I'm working on, I'm a theorist, so I'm building models. The theory on IDO is published, and the theory on the Itaconate shunt is my current project. So we need experiments to test, and that's what's going on in experimental labs like Ron's and Chris's. So that's the relationship between the Itaconate shunt and the IDO metabolic trap.
Oh, thank you. Well, I'm really grateful to you for doing this, and it just makes me realize again why nobody's really figured this disease out. It's so complicated, you know.
We constantly say to one another, this is the most complicated scientific project we've ever worked on.
And, you know, having sophisticated modelers and biochemists and et cetera working on this is such a gift to the ME/CFS community, because I don't really think we're going to get anywhere if we keep measuring the symptoms, which is what a lot of the research is doing. Now, of course, that's important because you have to know what you're aiming at when you're doing the models, right?
Yeah, every model I build is guided in part
Yes.
by my knowledge of what symptoms we have. You heard me talk about
Brain fog,
and PEM today.
Yes.
You have to have those in mind, because any theory, this goes back to David Bell. If you read David Bell's book, which is called The Doctor's Guide to Chronic Fatigue Syndrome, he says very explicitly that any theory has to account for 10 things. And I try to keep that list of 10 things in my mind all the time when I'm developing theories.
Well, this just makes me really hopeful. And just so you all know, Rob and Anne and Ron and I are going to travel around the world and meet the patients when they start getting better. And we're real excited about the prospect of being able to do that.
Yes, we are.
Okay. Thank you so much, Rob. Did you have any final thing you wanted to add?
No, I've already said it. Models and experiment. That's what we're doing. We're doing theory and practice.
Well, this
and
is.
I would say that all those people collecting massive data are doing science with just a slightly different perspective. They're saying, we don't know anything. Let's measure everything. And that's a rational approach. There are many scientists who have done it that way throughout
Darwin
right.
did it that way.
Exactly. So this is, you need both approaches. It's just that I'm a hypothesis tester. And so this is the way I work. I work from theories toward data and seeing if the theories can account for the data.
That's great.
Okay.
We'll
See you,
see you in
Janet.
part 2, and I'll talk to you offline about doing that like tomorrow.
Okay. See ya.
Thank you so much.